Okay guys, we are finishing up with genetic technologies now. I'm giving you a review note. This is the note that I gave out, I think it was first semester this year. I just wanted to put everything together for you so that as you leave genetics behind, you have some idea of how all this stuff fits together. So the note is titled Summary of Genetic Technologies. Okay, so that shouldn't have an S there. Summary of Genetic Technologies. And so what I really did start you off with was restriction enzymes. Okay, so you're writing restriction enzymes. What do restriction enzymes do? They cut DNA at a recognition site. Okay, and they make fragment or restriction fragments. So that goes back to the first lesson I gave you in genetic technologies. Restriction enzymes cut DNA. They were discovered in bacteria that were cutting the bacteriophage apart. They cut out a restriction site and they make fragments of DNA. So what was the original purpose? Again, the purpose is because this would disable the viral DNA and the bacteria wouldn't get sick. Okay. What are the bacteria looking for? They're looking for a target gene. So they're looking for a place to cut. Okay. And that place to cut is the recognition site. We are looking for a target gene. So I said looking for a target gene when we use these guys. Okay, so when we're using them. All right, now, plasmids. These are the circular form of DNA found in bacteria. This is where we insert our target gene. Okay, so whatever gene we want to have copied over and over again, that's the target gene, and we put them into a plasmid. Okay, remember the plasmid looks like this. I don't think you're going to forget because you just had that plasmid map assignment that you had to do. All right. Okay, so then we have restriction maps, which is exactly what I just said with the, sorry, I'm hoping you can see this. Okay. Restriction maps are what I just call the plasmid map. So it's a map of the plasmids. It shows the locations of all your known recognition sites. Okay, so remember I showed you the recognition site for E. coli, and the recognition site for E. coli, EcoR1, is GAATTC. So we have restriction enzymes, they cut at a recognition site. Okay, we look for a target gene, we put that target gene into a plasmid. Okay, and then we look to see if that's been copied in the plasmid map or the restriction map. So the restriction map shows the locations of all the known recognition sites. Okay, and so we know the recognition sites for all types of species, BAM, HI, HindI3. We have, like again, EcoR1. So you guys have looked at all those different guys. So restriction maps show locations of all known recognition sites. If we increase cuts, we increase the number of genes that we can insert into the plasmid. Okay, so if we increase the cuts, we increase the number of genes we can insert into the plasmid. And then moving into what you saw a day or two ago is PCR. Okay, polymerase chain reaction. How does PCR fit into all of this? This is how we make the DNA that we want to insert. Okay, so PCR is a method to amplify DNA, which means make lots of DNA. We run it on a gel. Okay, we can run our DNA on a gel for forensics. Okay, and for various other applications that I showed you on that note. And essentially, this is the DNA that we're putting into the plasmid. Okay, so we've got this plasmid. I said we insert a target gene. The target gene is this DNA that we have amplified. So we put this amplified DNA into a plasmid, and that's called genetic engineering when we do that. Okay, so we can run it on a gel, we can see it, we can see if we've amplified enough of it, and we can alternatively put it into a plasmid. When we put it into a plasmid and have the bacteria do the work for us, or another species, that's called genetic engineering. Okay? And then lastly, quite a few days ago now, I believe it was Tuesday, I showed you gel electrophoresis. So how gel electrophoresis works is you can run your amplified DNA, and it's actually a way to check to see, did you actually get the DNA that you wanted to make, okay, and did you make enough of it? All right, so this is a way of proofreading kind of, or checking, did your PCR work? Did your techniques actually work? So we run amplified DNA in an agarose, A-G-A-R-O-S-E gel, in an agarose gel, and separate it, okay? And how do we separate it? We separate it based on fragment length, okay? And how did we get those fragments? We used restriction enzymes. Alternatively, I showed you something yesterday where you can use CRISPR also. CRISPR, okay? So CRISPR is another technique used to cut DNA. It's widely used, okay? And it serves a very similar purpose, okay? So I just wanted you to be familiar with CRISPR, to have seen it. Basically, that's my goal. I wanted you to have seen it and not left for you without seeing it, so that next year in the event your prof say, CRISPR, you're not like, what the heck is that, okay? You have some idea that it's used to cut DNA.